Imagine the ability to cure genetic disease for generations to come, to inoculate the human race against the next COVID-19 before it becomes a pandemic, or in a darker scenario, to choose the color of your baby's skin. Dr. Jennifer Doudna pioneered a technology that may one day be able to do just that, one of the biggest scientific breakthroughs of our lifetimes. It's called CRISPR, a bacterial defense system that can edit genetic material. It already shows promise in eradicating malaria mosquitoes, appearing to cure patients with sickle cell anemia, improving cancer therapy, and diagnosing COVID-19 more quickly. And at the height of the pandemic in 2020, Doudna, along with Emmanuelle Charpentier, won the Nobel Prize in Chemistry for their CRISPR innovations. But there are major ethical questions looming. How and when is it right to edit a gene? Is CRISPR playing God? One Chinese scientist claims he's already edited the genes of twin girls, giving them immunity to HIV and sparking an international uproar. Right or wrong, one thing is clear: CRISPR will change the human race forever. Joining me on this edition of Bloomberg Studio 1.0, biochemist and CRISPR co-inventor, Jennifer Doudna. You grew up in Hawaii, and I am so curious. I grew up in Hawaii as well. How did your upbringing shape your curiosity about the natural world and the origins of life?
When I think back on that time in my life, it was kind of a wonderland. You know well what it's like in Hawaii. It's a very special place, and I found myself just wondering, how is it that these plants and animals have evolved to be specialized for Hawaii? And there are so many examples of that in the natural environments in the islands there. So I definitely think that was behind my thinking about, why it would be interesting to become a scientist in the future.
As a budding scientist, you developed an early fascination with RNA well before it took center stage in the pandemic, and also now a key player in vaccines. What is it about RNA that gripped your attention?
When I was an undergraduate in college, we were, or at least I felt like the message we received was that RNA was kind of the boring intermediary between DNA, which encodes all genetic information in cells, and proteins that do the work in the cell. And so those were kind of the important big molecules in biology. And then there was this kind of boring intermediary called RNA. However, when I got to graduate school, I met the person who would become my future advisor, Jack Szostak, and he was fascinated by the possibility that RNA, in fact, was the original biological molecule on Earth, that it was really responsible for the evolution of life as we know it here on Earth. And that idea was so interesting and so compelling to me that I joined his lab and I started studying RNA, and I sort of never left it.
Now, you spent most of your career working at universities. You're joining us now from Berkeley, where there's a lab named after you. But you briefly ventured into the corporate biotech world in 2009 and worked at Genentech for just 2 months. Why was that so brief, and what did you learn?
It was a really important experience for me, I have to say, even though short. Because at the time, I had been running my academic research lab for close to 15 years. And I was starting to question, what the impact of my work would really be. Was I actually going to, at the end of my career, feel that I had contributed to solving real-world problems? And so that was a big motivation for me to join the team at Genentech, a wonderful company where I knew a number of the scientists and some of the leadership team. However, once I got to the company, I quickly realized that I really missed my colleagues at Berkeley. I missed the academic environment of, being able to just think crazy ideas and, run into people in the coffee line and chat about experiments in a way that would be very difficult to do in a company, of course, because you have to be very focused on, the plans and development pipeline of the company. Fortunately, my colleagues at Berkeley took me back, and I refocused my efforts at the time on studying CRISPR, which in the end was, turned out to be a very productive line of work.
It's been called molecular scissors, if you will. What is CRISPR, and what can it do?
It turned out that CRISPR is in fact a system in bacteria that detects and cuts virus genetic material, whether it's DNA or RNA. And it was by studying how that actually works, and we did this in collaboration with Emmanuel Charpentier's lab, to study the function of a protein known as CRISPR-Cas9. That line of research led to an understanding of the function of this molecule that allowed us to harness it as a tool for genetic manipulation, namely for altering DNA sequences in any cell in a precise fashion, in a programmable fashion that gives scientists now a very powerful way to understand the function of genes, but importantly also to change the function of genes in a targeted way.
When did you realize the power and usefulness of this discovery?
Well, I would say almost right away. I mean, it's a relatively simple technology that can be easily adapted and adopted to different applications. And so very quickly after we published our work in the summer of 2012, labs around the world began to adapt CRISPR for various kinds of genome editing, and it's just increased since then.
The pace of CRISPR research, the application has been startling. It's been incredible to watch. What are the current use cases that inspire you the most?
Well, I always think about Victoria Gray, who was the first patient with sickle cell disease to be treated with CRISPR here in the US. Her story is so inspiring. I mean, she is somebody who is benefiting right now from the CRISPR technology to be able to live a normal life without being impacted by an otherwise quite devastating genetic disease. And other patients are similarly being impacted by the CRISPR technology. So I think that's one area where we'll see increasing developments, more and more clinical trials that are starting. In fact, at the Innovative Genomics Institute that I started a few years ago here in the Bay Area, we have just received approval from the Food and Drug Administration for our own investigational new drug, or IND, trial for sickle cell disease. So, this is really an extraordinary moment, I think, in terms of thinking about cures for genetic disorders.
Once CRISPR was confirmed as a gene editing tool, leading researchers raced to start their own companies, and it turned into kind of a competitive free-for-all. And the battle for the intellectual property is still going on to determine, who can commercialize this technology. How do you reflect on all of that?
One thing important to point out is that despite the ongoing disputes over patents, which, by the way, isn't unique to CRISPR, I would argue that any really exciting technology is going to have multiple claims to it. In the case of CRISPR, because the technology is relatively straightforward to deploy, it's meant that the field has moved quickly. As you mentioned, there are multiple companies. There are multiple companies that are now publicly traded and more coming down the pike. As well as all sorts of new companies and then established firms that are adopting the technology as well. So from a scientific perspective, I think that's exactly what should be happening, right? This is such an enabling technology. You want it to see it deployed as widely as possible.
What was your first thought when you heard that news?
I guess it wasn't entirely unexpected that someone would try to do this. I had no idea that it would happen as soon as it did.
At what point in your research and discovery of CRISPR did you start becoming worried about the ethical implications?
Well, quite early on, because it was clear from those very early days that CRISPR was a broadly enabling technology that was useful and worked in essentially any cell type, and that meant that it worked not only in fully developed differentiated cells or tissues, but it could also be used in embryos. And in fact, that was one of the very early uses in the research world, was to make modified mice at the embryo level. So those mice then had genetic changes introduced by CRISPR that could be passed on to future generations. And it didn't take too much of a stretch to think about the possibility that could also be done in human embryos, which of course I think comes along with just very profound ethical questions. I think it was 2015, we organized the first meeting out here in California on the topic of human germline editing, human embryo editing with CRISPR. And that developed into a much broader international effort to understand the technology and, importantly, to put in place criteria that scientists globally should respect in terms of applications of CRISPR, especially in the human germline.
Let's talk a little bit more about the reasons. If and when to edit genes is a profound and complicated question. How do you even begin to answer that question?
I think, first of all, one has to ask, are there situations where, at least in principle, manipulating the human germlines in the embryo would be the best possible way to deal with a genetic condition? And by the way, I'm focused here on, just really strictly things that relate directly to health rather than changes that might be, desirable to somebody for some reason, but actually have no benefit to health. And right now we know that although, yes, it can be used in human embryos, and there are, multiple scientific publications about that, we also know that it's difficult to control it and to make sure that editing is happening exactly as the scientist or experimenter might be desiring. And so that, to me, is already a red flag that, even if there were situations where we said, gee, that might be the best way to deal with a disease, the technology still needs to be further developed before that would be, I think, even a possible strategy.
Every new gene editing technology has its sort of cultural shocker moment. You had the first test tube baby, Dolly the sheep, and then, of course, in 2018, the so-called designer babies, where twin girls, their genes, were allegedly edited by a Chinese scientist, He Jiankui. What was your first thought when you heard that news?
Shock, for sure, definitely. I guess it wasn't entirely unexpected that someone would try to do this. I had no idea that it would happen as soon as it did, but, it had been discussed at meetings, of course, and that was, in fact, the purpose of these prior, conferences on the topic. So it seemed, it certainly seemed possible that someone would do this. I didn't think someone would actually proceed, however, to actually create a pregnancy with edited embryos, as was announced in 2018. And I think it really was a wake-up call to the international community that we can't sit back and just say, well, that's a problem for the future. No, this is something we need to deal with right now. And we need to take a strong stand. And I think, fortunately, that's exactly what happened.
Are other scientists trying these things elsewhere?
We really haven't heard about that kind of manipulation going on, certainly in any organized way. So I just think that, at least I hope, that the international reaction, which was really negative to this 2018 announcement, has I think, at least for the time being, really put a damper on anyone that might be trying to do that kind of human manipulation for, fame, for example. I do feel an ongoing sense that we need to be really proactive about this and not get complacent. And importantly, I include in sort of ethical considerations also thinking about widespread availability and affordability of the technology, because I think, this is something that we have to pay attention to. I mean, just thinking back to the example of Victoria Gray, who I mentioned earlier, who has received a CRISPR treatment for her sickle cell disease. Wonderful, news about that. However, her therapy currently costs $2 million. So, that's just not going to be affordable to most people around the globe that might need this. So we're working hard to think about ways that we can mitigate those costs.
Wanting to eliminate a genetic disease seems like an important cause. Do you think the moral case to eliminate a genetic disease is stronger than the case not to?
I think we have to consider it, on a case-by-case basis. I mean, one could argue, for example, let's go back to sickle cell disease, that, right now the therapy is designed to be used in individuals, and it doesn't make a germline change, right? It's not a change that they would pass on to children. It's just a change that affects their body. And so in that sense, it's like any other type of therapy or drug that we might use to treat disease. Whereas you could imagine that in a family that has a genetic disease that is sort of, spread across their family, many people inheriting a gene that, predisposes them to disease. And believe me, I hear from families like that almost daily. You could imagine that, at some point in the future, if the CRISPR technology were safe and robust, that some families might make the decision to remove that disease-causing mutation at the source so that, future generations don't have to worry about it. And I think that's, that would make a lot of sense. But again, there's a number of things that have to happen, I think, before that will be possible.
Back in the 70s, test tube babies were controversial, and now IVF is widely accepted, available. Do you think it will be the same with CRISPR-edited babies?
Over the coming decades, I think, sure. I think absolutely, that because this is what happens, isn't it? Is that, people get comfortable with an idea if it's useful, if it is proven, and this is still remains to be seen. Like if CRISPR proves to be useful and kind of controllable in human embryos, and that is still in the realm of research. But if that were to happen, then I think it becomes a possibility that in vitro fertilization clinics offer that to their clients. And so then, who should make that decision? Should governments be regulating that? Well, that doesn't really happen for IVF clinics right now. In fact, there's, as you probably know, there's, different regulations across different states in the U.S. and of course in different countries is different. And so I think, the same thing could possibly happen with CRISPR, where, it becomes something that, some clinics offer it and parents will have to decide, do I want to do that or not? When new technologies come along, they enable new science. When new science is done, they enable new technologies.
What do you say to the skeptics who say that CRISPR is playing God? The ethical controversies around CRISPR came screeching to a halt when the pandemic hit. What was going through your mind when the world met COVID-19?
Well, I think like many people, it was quite a shock. This was something that we really had to face head on. And that was actually, for me as a scientist, really a motivation for pivoting the focus of our work, at least over the last year, to creating a clinical testing lab at the Innovative Genomics Institute, and also to advancing CRISPR as a diagnostic technology.
You've had this fascination with RNA for so long, and did you ever think that your RNA specialty would have a moment like this, the key to fighting a global pandemic?
Well, no, I never thought that, for sure. And let me just point out, since you brought up RNA, I think it's fascinating that, first of all, the coronavirus, the SARS-CoV-2 virus that causes COVID-19, is an RNA virus. So it's a piece of RNA that's, been causing all this havoc. And furthermore, many of us have received vaccinations with RNA, right? So I received a vaccination that is a messenger RNA that carries a message into human cells telling them, make antibodies against this virus protein. And so that's been also very interesting. So RNA to fight RNA. And then with CRISPR, we have a third type of RNA that potentially could be useful, I think, primarily in this case, as a diagnostic tool, a way to detect the coronavirus RNA and report on its presence.
Couldn't CRISPR one day also be used to edit our genes to make us less susceptible to viruses?
Well, maybe. I mean, there's definitely speculation about that. Would it be possible to, I think about it sort of like genetic vaccination, in the sense that, could we educate ourselves ahead of time to be, ready in case a virus shows up? And it's a tricky thing, right? Because, you have to kind of know what to be ready for. And so, there would have to be some interplay there. But I think already we're seeing opportunities to use CRISPR to edit immune cells, and that this is being done in conjunction with cancer immunotherapy for patients, for example. So you could imagine taking that a step farther and saying, well, can we educate our immune cells to be ready for a virus?
What do you say to the skeptics who say that CRISPR is playing God?
Well, I guess my answer there is that, there are I don't even know how one defines playing God, because, there's so many things, ways that we manipulate our environment now. For example, all the food we eat is essentially engineered because of, plant breeding that's been going on for, thousands of years, really. And so when new technologies come along, they enable new science. When new science is done, they enable new technologies, et cetera. And in the end, that's really what drives human advancement, and it drives our economy in many ways. So I feel that overall this is all positive, but scientists really need to be engaged in accepting that responsibility for what they're doing and making sure that they're involved in the discussions and the decisions that have to be made as technology advances, and that's certainly true for CRISPR.
Let's go ahead another 100 years. If the next COVID-19 happens in another century, how will things be different? How will we be more prepared? Will our genes have already been edited to prevent us from getting the next big deadly disease?
I wonder. I guess I imagine that certainly within 100 years, we will know so much more about our own genomes. And the more we learn, honestly, the more complicated it clearly is. But so there'll be plenty to keep all of us busy. But look, I think in 100 years, we will know so much more about our own genetics, including the genetics of our immune systems and our interactions with viruses. So my hope would certainly be that, if the next 100-year pandemic comes along, that we will be certainly much better prepared to manage it than we were for this one.
So in the middle of this global pandemic, in October 2020, news came in that you won the Nobel Prize for CRISPR, which you shared with Emmanuelle Charpentier. 2 women winning the Nobel Prize. Looking back on those days when you were told girls don't do chemistry, what do you have to say to inspire the girls out there who might want to follow in your footsteps?
Well, it's, just such a kind of humbling experience for me in a way, because I certainly never ever in a million years would have imagined that, I would have won the Nobel Prize. And I'm even sort of shocked hearing myself say it right now. But here's an interesting thing that's happened. And that is that I've heard from many, many girls and women since then, some of whom I knew from my past life, but many of whom I don't know, who have reached out from all over the world to tell me their stories, to tell me that our work is inspiring to them. And I really hope that that message gets through very clearly to students, especially to women or other people who feel maybe they have been excluded in some ways from, or just been underrepresented in the STEM fields. That, I certainly didn't I'm not a likely person to have won the Nobel in a way, because I came from a small town. Nobody in my family was a scientist. I just kind of wanted to do science. I thought it was cool. And that's the message I try to tell those students. You can do this.
Well, thank you for paving the way for women and for all of the girls who will come after. And congratulations. Dr. Jennifer Doudna, thank you so much for joining us on this edition of Studio 1.0. It's been wonderful to have you.